Hi there, this is a review of Module 18, Look. So this is going to be about vision. Well, let's first talk about waves. So waves, we know that there's waves of light, there's waves of sound, but we're obviously talking about light right now. And when you look at a wave, the wavelength is peak to peak or low to low. I guess it's really trough to trough, whatever. So crest and trough, I don't know. But anyways, so there you go. That's what a wavelength is. And there is hue that's determined by that. The hue is basically just color. So wavelength can be in nm. That's the unit for it. And as you can see, there's a bigger wavelength for red, but then when you go all the way up to purple, it's smaller. Intensity. Well, this goes for both. This is determined by amplitude, so how crazy it is. That's what I like to say. So you see this thing, oh, kind of small. So that's going to be something that's not very bright or sound that is not loud, but then, whoa! This one down here, you're either going to go blind or deaf pretty much by that. So that's what intensity or amplitude is. All right, so here's the eye labeled. We see the different things. Let's talk about what they are. So cornea, we see this, that's this thing right here. It protects the light and bends it. So you're gonna want to know what all these things do. Pupil, that's the thing that's black in your eye, and it's the adjustable opening. So it gets bigger when it's dark, smaller when it's bright. It also can get bigger if you see something that you're interested in or afraid of. So if you saw something like that you didn't know what it was, like, I don't know what's a good example, but maybe if you're like watching a horror movie or something, you probably have bigger pupils because you're not knowing what's going to happen. But light enters right there. And then the iris, that is the colored part of your eye. So probably it's brown or blue. I think that's pretty much the only eye colors that people have. Green is pretty much, well, yeah, you can have green eyes too. This is the colored part that controls the size of the pupil. So it can make it smaller, it can make it bigger. Lens, that's something that you don't see, it's behind. And it helps focus the image. Then the retina, that's this thing back here. It's the thing that has receptors, rods and cones that help process the information. And so I didn't talk about these things yet, but I will. And it's something that might be good to know is that when there's an image going back here, it is upside down. The image that's cast in your eye is upside down. Oh, well, see, that's why the candle's upside down right here. It's upside down, but then once it gets to your frontal lobe, the optic nerve takes it to the brain's visual cortex. That's in the occipital lobe, right? Then it goes to the frontal lobe, and it's back to normal. Okay, so here's the order that it goes. So cornea, pupil, which is controlled by the iris, then it goes to the lens, then the retina, and then it's processed. So when it's processed, here's what happens. So it goes to your rods and cones, and your rods are basically what makes you see black and white, and they are used in more dark situations. And then the cones, those are for colors, and that's what you can see during the day. Then it goes to your bipolar cells, which are activated by the light energy that enters. And then these bipolar cells, they activate something called the ganglion cells. Then after that, it goes to the optic nerve. And like I said, then it goes to the visual cortex, which is part of the occipital lobe. Then it goes to the frontal lobe, and there you go. The fovea, this, you see right there, that's where it is. It's the central focal point of the retina. It's where the cones are. It helps with visual acuity, which is distinguishment. So the fovea it can help with like complex images. So it's like, what is that? Oh, fovea. I know ya from my fovea. Well, not really, because you interpret what something is from your brain, but it helps with distinguishment. So, all right, photoreceptors. What are these? These are neurons that are found in the retina that convert light into electrical signals that stimulate physiological processes. They relay information by bipolar and ganglion cells. So you see there's the photoreceptor, then it's transmitted to the bipolar cell, then transmitted to the ganglion cell. Then the optic nerve, I already said that. Seeing better at night. So you see this cats and other animals can see better at night because of their higher proportion of rods and cones. Okay, so feature detectors. What are these? These are things that help you see angles, edges, and movements. So if you didn't have good feature detectors, then this is what you might see. You know, I mean, can't really see that many angles or edges, but you can better for this one. Okay, parallel processing. What is this? This is just simply how we process vision. It's the visual system in the brain that divides what it sees into 4 components. These 4 components are color, motion, shape, and depth. They are individually analyzed and then compared to stored memories, which helps the brain identify what you're viewing. So it's like, all right, what did I just see? What have I seen? Oh, okay. That's the same thing. That's not the same thing. And then it determines what it is. Young-Helmholtz trichromatic 3-color theory. Tri means 3, right? This is the theory that there are 3 color receptors, red, green, and blue, that combine to produce other colors. So are you colorblind or cone blind? Cone blind isn't a real thing, but I'll tell you about that. It's, well, that's not a real term, I mean. So are you colorblind? Let's see. Can you see what number is right here? I can. It's 74. I am not colorblind. But over here, here's the problem. What color are these 2 things? You might say blue. I say purple. So this is just an interesting thing that has happened to me for a while, and people tell me that things are blue when they're purple. I've seen people wear shirts, and I said something about it being purple, and they're like, "What? That's blue." And I was at like Golden Corral one time a few years ago, and I was like, "Can you give me that purple plate?" And then people were like, "It's not purple, that's blue." So it's weird because it's not like I can't see blue. I definitely can. But anytime I see a shade like this, it literally looks purple to me. And I don't know, it's just a weird thing. But the reason why that's the case for me is that there's something with my cones that's different. Remember, the cones are what see color. So there must be some kind of cone issue that I have. So maybe there's other people that have the same thing. And it's not always just this. Occasionally I'll have the problem with distinguishing red and orange and green and blue, but typically it's blue and purple that's the problem. But okay, flag test. So what's the after image? This is if you stare at something, you can see the opposite colors when it's gone. So look at that red dot for 30 seconds. So pause the video and look at that for 30 seconds. Don't blink. Okay, assuming you already looked at it, I'm taking that away. You may see a red, white, and blue typical British flag. And that's because of the after image. Opponent process theory. This is the theory that opposing retinal processes, red, green, yellow, blue, white, black, enable color vision. And this is what proves this after image thing. Because if you notice what colors are on this thing, they are green, yellow, black. So green changes to red, yellow changes to blue, black changes to white. All right, the neural train. So what happens during the sensation? This is sort of has to do a little bit more with module 19. But nevertheless, there's still something good to know. This has to do more with feeling, but this might just be good to know. This wasn't actually in module 18, but this is kind of a good intro to the next module. So here's what happens. First, so let's pretend you're having a handshake or something. All right, you shake your hands with someone. There's going to be receptors. There's pain, temperature, and pressure. So what's going to happen? Let's think. Would that be pain, pressure, or temperature? Well, it could be any of them. Probably pressure. If they were squeezing too hard, it could be pain. If your hands are hot or something, then it could be temperature, but probably pressure. Then after that, then there's the sensory neurons, which are also called afferent. Remember, we learned about these in like module 9. And then the interneurons, they do the things in between. And the motor neurons, they're also called the efferent neurons. And then it goes to the muscles and glands, which are the effectors. Okay, Hubel's and Wiesel's study. These are two guys who figured out that cortical cells or cortex cells, they respond most strongly to specific visual information. So that means that the more specific something is, the more strong that the cortical cells will respond. That's what they figured out. Okay, what is a motion parallax? This is the idea that closer objects seem to pass more quickly than far away ones. So think about if you've ever been to an airport, you see a plane on the runway. Seems like it's going pretty fast, right? Well, it's going probably even faster when it's in the sky, but it looks like it's going slower. That's because it's far away. That's motion parallax. All right, let's look at some questions here. When are bipolar cells activated? They're activated when light energy triggers chemical changes that spark neural signals, activating them. Why can cats see better at night? Because they have a higher proportion of rods and cones. Where in the eye is visual acuity the best? That's in the fovea. What's the correct sequence of the neural chain of events set in motion by environmental stimuli? Remember, I told you that this was not really having to do with vision. These are kind of like these questions are like a guide for the module, but sometimes I already have said before, I don't make these. So sometimes there can be some kind of mix up. So this is really a module 19 thing, but it is receptors, afferent or sensory neurons, interneurons, efferent or motor neurons, and then effectors, which is muscles and glands. Explain Hubel's and Wiesel's study or research on responses to the brain. Remember, they figured out that cortical cells respond most strongly to strong visual information. How do photoreceptors relay visual information? They do that by bipolar and ganglion cells. Oh, not done, sorry. Provide the correct order of processing in vision. Remember, I told you the order. It's first the rods and cones, then bipolar cells, ganglion cells, and then the optic nerve. Explain the opponent process theory. This is the theory that 3 sets of retinal processes, red, green, yellow, blue, white, black, enable our color vision. Why do nearby objects seem to pass more quickly? Sorry, than objects in the distance. That's due to motion parallax. Why would a person have difficulty with visual perception after a corneal implant? This is kind of like a multi-step thing or something. Think about it. What does the cornea do? It protects the eye, and it bends light. So since cornea provides protection and focus, an implant would make perception difficult at first. Which theory explains after images? That's the opponent process theory. What's the purpose of the iris? Remember, it changes the size of the pupil opening. Would a feature detectors detect angles, shapes, and environment? Well, that's it. Thank you for watching.